like to acknowledge that we here at the University of Alberta campus are located on Treaty 6 territory and respect the histories, languages, and cultures of our First Nations, Métis, Inuit, and all First Peoples of Canada, and are committed to reconciliation, ending systemic racism in partnership with our Indigenous colleagues. Welcome to ATI rounds on November 6, 2024. And thank you very much to our sponsor, Paladin. And I really, it's a great pleasure to welcome a very well-known colleague and a friend from our Western province and a partner in our transplant programs, Dr. Paul Keown, who doesn't really need a lot of introduction. He is a very well-known individual in the national and international transplant community and is a specialist in immunology and transplantation, a professor with UBC in the Department of Medicine, and is a practicing physician at the Vancouver General Hospital. He has served in many leadership programs provincially and nationally and internationally, and has been a major impact, has had a major impact on how we do HLA testing, actually, and is still advancing the field very actively, most recently with securing large-scale investments from Genome Canada to take HLA from the, I think, what was it? Pure cells in the past, and then we went to flow, and then we had artificial or structured beads, selective, and now Paul is taking it to the next level of high-resolution testing using molecular methods and standardizing this at a national level with a direct impact on how we care for our patients and our national exchange programs. And it is a great pleasure, Paul, that you could find the time today to be here and give us an update of this national work, which we're also here in Alberta part of, and which I think is very exciting and a true practice and game changer. Thank you, Paul.
So we started many years ago, probably in the just about 2014, thinking, how can we make the next change in transplantation? So we've come a long way over the past 30 or 40 years. So Kim Soles, Phil, yourself, Michael, and a few others remember the old days when we started in transplantation in Canada, when within the first year, 80% of people had very severe rejection, really blowout rejection. 50% of grafts were lost and 25% of patients died. It was a really, really challenging period. And then we have come through to the period of about 10 years ago, where rejection rates had fallen to maybe 15%. Antibody-mediated rejection, still a major problem, but vastly better than it was. So the question at the time was, could we bring together a consortium to work on the individual problems to move us from there slowly to the point where we would have very little rejection, and we could begin to think seriously about the question of induction of tolerance, which had until now only been a dream. So we formed the Genome Canada Transplant Consortium. You will see many of the universities listed here. There are now about 27 universities who participate across Canada, in the United States and in Europe, and about 70 PIs who are involved. And the leaders in our site here, apart from myself, are James Land, who now runs the immunology lab, and Karen Sherwood, who is one of the associate directors in the lab. So as we were thinking of the first Genome Canada proposal, how to phrase it, put it together, we were struck by the tremendous article that came out from yourselves in Alberta in 2013, looking at the causes of graft loss. And really, in a nutshell, it said that the major cause of graft loss was rejection. And within that, which affected about 60% of the failed grafts, the leading individual type was antibody-mediated rejection. So we phrased the first Genome Canada proposal, which is just coming to an end, ran for 7 years, as a way to prevent antibody-mediated rejection. So to us, the goal was, first of all, to prevent crossing antibodies, and the second was to begin to match more effectively. But at this time, we looked at the long-term outcomes, and this is some work from Adrian Levy, who was one of our senior health economists at that time, who working with the US data showed that by the time you get to 15 years, even in patients with very good GFR, half the grafts are lost. And of course, that time reduces further and further if they start out with poorer graft function. And patient survival falls at the same time. So we had two problems. One is early problem, and the second is progressive, unstoppable graft failure. Bringing the Genome Canada program together, I mentioned 70 principal investigators from, at that time, 22. Now we've increased it to about 27, and we've added on countries like Australia. We began to build a consortium that would really reach across all sorts of borders. So we started out with the regulatory agencies, Health Canada, the FDA, the organizational groups and clinical programs across Canada, all of the 13 HLA laboratories, and then a whole series of other proteomics laboratories, immune laboratories, data sciences groups, and other research partners. And then we turned to network partners, largely from industry, who worked in the fields of innovator companies, global academic experts, and healthcare users and providers, many of the clinical teams across the country, but also including payers, healthcare providers, patients, and caregivers. The goal was to try to develop a holistic network that would help us to move forward. We drew very heavily on the resources of CBS at the time. We drew on their regional laboratories, 13 laboratories all across the country, the regional transplant centers, just over 20 of them across the country, and on the provincial programs, the national programs, and many support services, which they provide or help us to provide across the country. And we developed 4 pillars for the first genome program. The first was to optimize matching. The second was to personalize therapy. The third was to develop informative post-transplant surveillance. And the fourth was, which is required by Genome Canada, or strongly suggested by Genome Canada, was to develop evidence-based health policy so that we can engage not just the providers of healthcare, but the receivers of healthcare. And we embraced all of this under the heading of precision medicine. So let's start with number 1. Here we began to think seriously about the way we match. And I'll show you that the conventional methods of matching in the past are completely impossible because of the enormous variability of the HLA region in genotypic and proteomic structures. So our goal was to understand the structural biology that might take us forward to epitope matching, then to develop methods that would allow sequencing rapidly, much more rapidly than we were doing at the time, and then to develop a potential Canadian program for epitope matching. Now, we forget, because today we're so advanced in terms of the technology of HLA, that going back even a decade, it was a very different landscape. We were using relatively simple SSO and SSP technologies for typing. They had ambiguities. They weren't necessarily quick. We had to repeat typing. It was really a very challenging area to begin to move from at that time. There were some which were really interesting and really fast, such as linkage bioscience, at the time the biosciences, but now been acquired by Thermo Fisher, which was fast and simple, but contained ambiguities, and the imputation from it was not close to 100%. So these are all great if you're typing recipients because you have plenty of time. You can retype, you can be sure. But if you have a donor and you have 4 or 5 or 6 hours to type them, then it's very difficult to get sequence level information with confidence using the older approaches. The challenge is shown here. When I first came to BC in 1987, the number of alleles for HLA was very close to 0. You can see almost none way down here on the left. And then over time, the number of alleles has risen so that today It's much more than this. It's over 38,000 alleles from the 11 conventional genes that today we sequence. So clearly with 38,000 alleles, there's no possibility of doing meaningful HLA matching between donor and recipient. We need a different way to look at this. But as it turns out, much of the structure of the HLA protein is common and shared between all of us. There are only a few hotspots, which we call epitopes or eplets, depending on the terminology. Epitopes, usually the linear peptide for the sequence, and then an eplet being the configuration where the amino acids may not be in linear sequence, but may be adjacent so that they're recognized by the antibody. So now we began to look at the number of eplets, where they lie, how we could consider these as new opportunities for matching. And the person who really has done a tremendous amount of work on this and who deserves enormous credit is René Duquesnoy, who was in Pittsburgh for many years of his life and has just retired. So breaking down and mapping out these eplets, we can begin to see where they lie. Today, there are about 70-something class 1 eplets, and I'll tell you the numbers of the others in a moment. And the reason I'm hesitant is because not all of them are antibody verified. Many of them are old legacy eplets, and we're trying to define them a little more clearly. But for today, we'll leave it very simple. So these are the different HLA genes and HLA proteins in class 1A, B, and C. And you can see that many of the eplets are shared across these class 1 molecules. Whereas when we move to class 2, DQ beta, DP beta, DP alpha, DR beta 1, 2, and 3, the eplets are constrained within each gene locus, or the products of each gene locus. So there are now perhaps in the region of 36 recognized DR beta eplets, about 51 at DQ, only about 11 at DP. And a lot of information tells us now that these are much more important than the class 1 eplets in terms of matching. So our first step was to ask, what do we see in a population? We took the BC population, about 1,000 patients and donors here, and we sequenced them all using next generation sequencing. And from the sequencing, we could identify with confidence the alleles at class 1, class 2, and then we could begin to break them down into the individual eplets. And the eplets are down here, the alleles are up here. So you see, we reduce the complexity as we move from alleles to eplets for both class 1 and class 2. We're moving from somewhere in the region of 400 or 500 alleles, which are identified within BC, to a much smaller number of eplets. It's surprising that of the 38,000 known alleles in HLA, only a few hundred are actually expressed in our population. And you all know that BC is a very heterogeneous genetic population, but even so, most of the alleles in HLA are rare alleles. So now another thing became very interesting to us. This is the frequency. The top two bars are the frequencies of the class 1 and the class 2 alleles in British Columbia. And you can see there are some common alleles. This is about 40%. And then at class 2, this may be about 60 or 70%. But the frequency declines very quickly, so that the vast majority of alleles we identify are seen in 1% of the population, and the same in class 2. Whereas when we translate those to eplets, the linear relationship is much more standard across the population. Yes, it goes from most common to least common, but it's not a rapid decline to almost zero. Most of the eplets are expressed much more frequently across the BC population. So now we can turn to our retrospective cohort. We have a very nice retrospective cohort extending back over 30 years in BC from the time I first came. We can take all of the data on those patients who were transplanted over that time. We can sequence them, and we can ask two questions. First, what was the probability of them matching just by chance alone at the eplet level. So we can match them for all genes, or for DR beta 1, 3, 4, 5, or for class 2, or for DQ, or any combination we want. The black line shows what happened in real life. So let's take just DQ beta. In fact, this is the mismatch score, the number of eplets mismatched. And you can see that if we look at 50% of patients, the mismatch score is about 5. Not huge, but still substantial. And if we look at others, for example, class 1, you see the observed frequencies of mismatch, and the same for the class 2 overall here. Now we can simulate the data. We can say, if in fact we had the same donors and recipients, but we had actually taken the donors and given them to the best matched recipient in epitope terms, how closely would we have been able to match them? So we're now asking a question of feasibility. We're asking, is it feasible to achieve a high proportion of patients who would in fact receive a well-matched donor organ? Now we can look at the red lines. So if we look at DQ beta and we ask, if we wanted to match only for DQ beta, what's the probability in this same population that we would have been able to do so? And the answer is that over 90% of the patients would have been matchable at DQ. And if we want to use combinations of DR beta 1, 3, 4, 5 plus DQ, you can see it's still very good. And even class 1 and class 2 improve. So we then concluded that actually this is feasible. First of all, we reduce the complexity so we have fewer targets to match. And now if we allocate them in a real population, this is what we would hope to see. To make things simple, I'll mention that we have begun to focus increasingly on DQ, because the growing evidence suggests that DQ is in fact the most important driver for us. The bottom line shows the probability of matching over time. And to the right-hand side are a number of red individuals. You can see the slope rises slowly, but then takes off. And these signify the individuals who would be more difficult to match. So it suggests we can actually identify a priori when these patients first come for a transplant assessment, who will be the difficult ones to match. And then we can think about approaching them in a different way. So here we have again the same approach, but with one thing added. If we think about adding a donor into a waiting list, if we only have 10 people on a waiting list, let's say like a heart transplant population or a lung transplant population, the chance of finding a really good match is not so high. If we have 1,000 patients on the list, then the chance of finding a good match is really high. So the probability of matching depends upon the size of the donor pool and the donor to recipient ratio. So what we have done now is modified that simulation to look at the wait list sizes across Canada. And it goes from 100 patients on a wait list to 2,000, which would be the whole Canadian waiting population. And now we can look at each of the genes individually, class 1, class 2. This top one is DQ beta. And we can see that the probability of matching increases as the waiting list grows up to a certain point, let's say around 400. So if you have a waiting list of about 400, you have a 90% probability of achieving a DQ match, perfect DQ match in this situation. If you want to relax that a little bit and you're prepared to take a close but non-zero match, it's even better down here. So we can look at this and we can think of our own waiting list sizes. We were around 400 for many years, but due to the unfortunate drug death rate in BC, our waiting list has declined a little bit, so we're moving in this direction. The reason for this was to ask, if we were to move to epitope matching, could we do it within province? or do we have to share across Canada? It looks as though there's no advantage of sharing beyond, let's say, 800. So even at the very worst, we may consider pooling regions. So Alberta and BC could be one region. The Maritimes could be another region, and so on. So this provided the theoretical basis for epitope matching, but not the practical basis, because we need precise sequence information on these donors in order to be able to match them. So this is where we move to the new sequencing technology. When we first put forward the proposal in 2017, we proposed developing nanopore technology with a little company in Oxford Nanopore. And they were very promising, but their precision rates at that time were extremely poor. And in fact, one of the big questions that came to us as we went for the interview for the grant was, do you think this will work? And there was a lot of skepticism. But actually, it's worked tremendously well. And it's worked so well, I think it's going to replace the conventional short read sequencing, which we use everywhere. And these are the technologies. Actually, we can look way down here because this is the little one called the Minion, and it's about the size of a cell phone, maybe actually smaller than a cell phone. And you can read the data on your cell phone. So it's a transformational change in sequencing. And it will enable sequencing of all 11 HLA genes in a period now of about 4 hours. So it offers precision because the reading is long, and it offers speed because of the technology. So this is just some data to show you. The internal validation. These are our team who did it, and we pooled a whole series of samples. This is a slide kindly loaned by James. Proficiency testing samples, clinical samples, novel. We have just described 18 novel sequences which we've submitted to the database, rare, and then buccal swabs as well as blood. Buccal swabs are challenging to sequence because of a lot of reasons due to fragmentation of the DNA and other things. But you can see the accuracy here using nanopore technology is 100% for all of them. This is a wonderful technology. And we're very grateful to Karen over on the left. Many of you know Karen, who has done a lot of the foundational work in setting this up. You see here the Minion being loaded, so you actually appreciate the size of this technology. So then we went through all sorts of questions to validate it. Does it make a difference whether we have, what are the number of reads on the right-hand side that we get if we use blood, or if we use buccal swabs, if we use buccal with low yield? In fact, anything over about 5,000 reads for long-range reading is excellent. So we were very impressed that this is a very powerful technology. And we're able to compare conventional NGS with nanopore sequencing, and all of the problems that we saw with the original NGS, which is short read sequencing. So it's like building a brick wall. You put the bricks on top of one another, and that gives you your ultimate read. With nanopore sequencing, you actually have the long stretches of the wall, which you can put on top of one another. So you need far fewer of them. And we have 100% success with no ambiguities. The question then about timing, how quickly can we do this? And you see over here, run by Franz, in the range of about 3 hours, 50 minutes to 4 hours and a quarter. So clearly very suitable for donor time. Now we proceed to validation. So the process we've adopted is a formal health technology transfer evaluation for all of the technologies. One site serves as the developer site, another site serves as the validation site, and we exchange those roles throughout the consortium. For this one, we were the developer site, and McGill Genome Sciences Center was the validation site. And they achieved, of course, very similar results to the ones we did, so confirming that this is fine. Now, at the same time, we began to think of the question of perch. I'm sorry, I think I've just got the one reversed here. But let me tell you just the same. So perch is another way of looking at T-cell perceived epitopes on the graft side. And whether it has the same clinical implications, we're not truly sure, but it certainly is related in the same way to the probability of rejection. So we repeated the simulation. And in fact, if I told you that this was perch, it would be almost indistinguishable from the ones I've shown you for the B-cell epitopes. The probability of close matching is almost as good, not quite as good, but very close. It works most effectively with DQ perch, and it shows the same relationship to waiting list size. And when we simulate it, we see exactly the same thing. By chance alone, we would have this degree of compatibility, the perch scores down below. And if we actually deliberately allocated on the basis of DQ perch score, we would have this probability of matching. So a very, very close to 100% probability of matching. We can actually adjust these models now so we can take all of these questions into consideration. We can ask, if we change the donor population and the recipient population to be A and B, and if we look at the pool size, what do we get? And this is called a Sankey model, named after an engineer, Sankey, which shows us how the baseline can be transformed into the outcome depending on the criteria we choose. Let me move from the perch now. The other interesting feature was the figure I showed you before showing the difficult to match patients is almost identical with perch. And we can look here and we can see which are the ones who are going to be problematic. Move past this. So let me move now to personalized therapy. We have the possibility of implementing the sequencing across the country so that patients could, in fact, move into an epitope matching program across Canada. And the goal is to set up a series of trials. We've already started the phase 1 trial here. James has just reported actually some of the stunning data where our rejection rates have fallen to less than 1% with 2 antibody-mediated rejections in the first 1,000 patients. So dramatically impressive. We're now moving to the phase 2, phase 3 trial, with the phase 2 being started in BC, and the phase 3 will be run across the country. But now we need to think about therapy. So as we reduce the risk of rejection, it gives us an opportunity to adjust therapy. And it's very sobering to think that the drugs we use today are in fact over 30 years old. So many of the older members of our audience today were engaged in the early trials, the first pivotal trials of the drugs we use, tacrolimus, sirolimus, mycophenolate. Those are all drugs from that. They were tremendously transformative, but are highly toxic. And we want to minimize them effectively or get away from them. So I've used 2 terms so far. One is precision medicine, which for the purposes of today, I've intended to mean an understanding of the molecular causes of a disease or disorder and the ability to target those with therapy. And the second is personalized medicine, which means the application of this understanding to the genetic landscape of the recipient so that we can individualize treatment for that individual. So we're beginning to work on areas of systems pharmacology, novel clinical trials to evaluate treatment, and cellular therapeutics that we think will take us through to operational topics. So let me show you some of the data from some of James's trials. The green line at the top shows a retrospective cohort of close to 1,000 patients, identifying those who were epitope matched at the DQ locus alone by chance. And you can see that the risk of rejection is essentially 0 in the first year. This doesn't say that they will survive forever, because we're beginning to believe that the factors which drive early rejection and late rejection are different. For late rejection, we have non-adherence to medication, a whole series of other things. But just focusing on the early phase, we see that's the case. So the second kind of question James has been asking is, does it make a difference if we use different kinds of induction therapy in these populations? And In the interest of time, I will simply tell you that if you are closely matched, it makes no difference whether you have basiliximab or ATG for induction. But if you're not, it does make a substantial difference. So we're beginning to get an understanding of personalization. So this is the model that we've drawn up to begin to explore it more fully. First, we start with an epitope match. Either people are or are not epitope matched. And then based upon that, we have information from immune monitoring, which will give us an understanding of the activity of their immune response. For example, children have very active immune responses. Older individuals usually don't reject, and they die from the toxicity of the medications we give them. And together, we begin to personalize therapy based upon this. So we're just submitting a new Genome Canada application now that will look at deep genotyping of donors and recipients, probably about 4,000 of them, to begin to understand in greater detail the factors which relate to personalization of therapy. Now, if we want to do trials in transplantation, we have to abandon the old model. The old model was the very classical two-arm comparative trial, one experimental arm, one control arm, often large numbers of subjects, several hundred in each, and we wait for a couple of years to look at the outcomes. But that's far too slow for the speed of change which is occurring today, and it's wasteful of patients. So we want to begin to look at things in a different way. We've taken the experience of our oncology colleagues who have radically transformed things using rapid adaptive design trials with synthetic controls. So in essence, they have one experimental group, and all of their controls are synthetic controls from their pooled data set. So they may take one Mrs. Smith, who's a 50-year-old lady with diabetes and many other factors, and from their population, they will take 4 Mrs. Smiths who are identical or very close to the one who is in the experimental arm. And this means we can now adjust the sample size. So if we need a sample size of 200 subjects, maybe 50 of them need to be in the experimental arm and 150 can be in the control arm. It gives us an enormous amount of flexibility. And also we've changed the concept of these trials. We want to understand if some things are likely to work or not likely to work quickly and not waste a lot of time on those that are not likely to work. So we've shortened the duration of these trials and made them very focused. So we have 3 sets of synthetic controls. Site-level synthetic controls, for example, those would be controls from BC for our own patients. Trial-level synthetic controls, where we take them from reported trials, or population level, which might be we take all the population of patients in Canada and we distill those and we use them for each of our individual studies. And this is how it looks. We would have, for example, let's say we're putting 50 patients into a trial as an experimental group. We would look back to the simulated patient group and we would select the patients we want according to the group. These would be our actual patients. We would simulate the controls to match them, but now we can make them 2-to-1, 3-to-1, or 4-to-1. So we maintain the statistical power and precision, but with very smaller numbers exposed at risk in the experimental group. And if we now bring in automated stopping rules, we actually have an automated trial. We run it until we get the signal within a certain time. We move then to the next stage of the adaptation. So where are we going to go in the next step with this? Well, we think that if we are able to achieve epitope matching effectively and efficiently, which we believe is now possible, we should see the risk of rejection dramatically reduced. So the 2 I mentioned to you were the retrospective trial that James has run and the prospective phase 1, which he's just preparing to write up now. Both of them show that antibody-mediated rejection in the first year has almost disappeared, less than 2 cases in 1,000. And the risk of acute rejection for patients who are well matched, and now we can match, we believe, over 90% of them, the risk of acute biopsy-proven rejection is also less than 1%. So this transforms the scenario. It really means we go from a big storm on the West Coast here to quiet, beautiful summer's evening with a few ripples on the sea. And if that's the case, it resets the possibility for things like the induction of tolerance or partial tolerance, where we dramatically reduce the immunosuppression. So this is potential, but not easy to achieve, and we need to think very carefully about it. So the first step is, how do we adjust the immune suppression if their risk of rejection is very much reduced by epitope matching? So these are the short-term simulated studies that we're beginning to engage in now, looking at different combinations, looking at genetic control of the pharmacologic response through pharmacogenomic genes and a whole variety of other places to try and personalize the immunosuppression for each individual. And then, assuming we can bring the immune suppression to a certain level, we would now begin to look at the potential for induction of operational tolerance, for want of a better word, using the approaches that they've adopted in Northwestern, in Stanford, and a variety of others. And these have two components. One is the expansion of donor stem cells at the time of transplantation, which are given to the recipient sometime later, usually somewhere in the first 2 weeks later, and then solidification of this quiescence and microchimerism through expanded autologous regulatory cells. And you can see here that we're extremely fortunate. The Canadian government has invested very heavily in this over the last year or two, has donated $500 million to a series of 5 bioengineering hubs across the country, of which one is here, which is run by Megan Leving, who is a partner with us in the program. So this is really a very exciting opportunity. And having been involved in transplantation now for well over 40 years, I think this is the first time I feel that we are close to actually achieving operational tolerance. So I mentioned CBS in the beginning, and we've been working very closely with them. And what we would like to do is to set this up as a national program with the support of CBS. I use this just to give an example of the sort of program they've set up for our highly sensitized patients, for our paired exchange and others. And now we're working with them to begin to set up an epitope matching program so we can begin to pool our data. They can record, everyone then can see exactly how things are progressing in the genome program. Okay, let me turn now to the third one, which I think will become increasingly important. So let's assume that we're successful in the first steps. We can reduce immunosuppression. We have epitope match patients. British Columbia, as you know, is like your own province in Alberta. British Columbia is bigger than France and Germany combined, and we have a population of 5 million. So patients are distributed all over the place. They stay with us now for the first 4 or 5 weeks in Vancouver, and then they go home. And it's once they go home, they begin to enter into a potential danger zone, because when they're here, we look after them meticulously. When they go home, of course, quite rightly, they want more freedom and independence, but that's when things can go wrong. So what I think we're exchanging is intensive immunosuppression lifelong for much more cautious and careful and continuous monitoring of the immune response post-transplant. So we want to be able to monitor the response to the graft, post-transplant infections, and post-transplant cancer. We've done a lot of work on this. I'll only mention some of the areas. We've looked at deep phenotyping in a variety of ways using AI to integrate all of the information and to reduce the complexity of the data. We've done transcriptomic studies looking longitudinally at patients. Let me simply say that these are informative, but not answers, not predictive answers to the development of rejection. But what I'm very hopeful for is the use of T-cell receptor sequencing. So if we think empirically, the first response is really the recognition of the graft by the recipient T-cell and the expansion of the clones, which recognize those specific epitopes on the graft. We should see clones expand. We know that we do. And so we should be able to sequence the T-cell receptor genes. And if our hypothesis is right, we should see certain gene combinations grow in frequency, reflecting expansion of the population. And we do mixed lymphocyte cultures before people are transplanted. And you can see here two populations, very discrete, very clear populations emerging after transplant. We have to do a lot more work on this, and it's going to fit in with our ongoing studies at the moment. So many of these processes are actually all integrated and built through our current and our next genome grant. But let me turn away from immune monitoring to some new work we've been doing, which is in viral infections. So we know that viral infections destabilize the recipient post-transplant. And in association with them and with their treatment, we see profound leukopenia. Often that causes us to reduce the mycophenolate, and then rejection occurs. So it's not a simple problem. We've applied a lot of the learning from COVID to beginning to explore CMV. So the first question is everybody at the same risk? And we think not. And we think that your susceptibility to to CMV and its long-term complications may be determined by your HLA configuration. So we know that some HLA molecules are wide open and permissive. They will accept almost any peptide. Some molecules are very narrow and restrictive and will only accept a few. And we begin to see that our HLA type is probably closely related to the ability to present the MHC peptides. And so we believe the potential for us to respond to them. We're testing that further. The second thing of interest was, does the understanding of the role of viral load kinetics, magnitude of the viral response, which emerged through COVID as a dominant factor determining death and illness, does that play a role in CMV? So we've gone back to our last 1,500 patients that were transplanted, actually a little bit more in this one, excuse me, we added the living donors to 2,500. You can see them here. These are all patients on whom we have full long-term follow-up for up to 13 years in this population. We can now see the frequency of first and multiple rejection episodes over time. The really busy period, of course, is the first 500 days. That's when most things occur. The probability of infection is determined by age. The older you are, much higher risk of infection is determined by whether your donor is positive and you are negative, and a whole other series of things. Did you have early graft function? Did you not? And so we can begin to determine composite risk scores, which will include genes, which will include other phenotypic factors. And we can now map the viral load throughout the whole of the viral infection course for each patient. So when they first develop viremia until the end of a viral episode. And we can put these together. If a person has 2, 3, or 4 viral episodes, we can now summate them and begin to look at the risk. We can see that the duration of a viral episode and the area under the curve for that viral load, for that viral episode, are very closely related. Duration, cumulative AUC, just as we would expect. And it gives us a rule of thumb. And the rule of thumb, in essence, says that if you have viremia for more than 15 days in your first episode, your risk of graft loss is significantly increased. And your risk of death is increased, though not quite significant. But then we go further and we can say, if you have more than one episode, then you're at particular risk. So first episode, second episode, third episode, fourth episode, this is the cumulative duration in days. And if you have more than one episode of CMV, you become at profound risk of graft loss and death. And we can build composite risk scores or composite risk pictures of these patients. And in general, they tend to be older. They tend to have poorer graft function. They have early delayed graft function. They have often received ATG. And interestingly, in our population, they tend to be non-Caucasian. So this stumped us for a little while in the beginning until we looked more closely into the genotyping of the CMV virus itself. And there are, in fact, a number of genotypes which come from different regions of the world. And it may well be that this is vitally important in determining outcome. And we have there's some very interesting recent data just come from I think the Utrecht group, which says that this may be important. This difference in genetic type between donor and recipient may be important in triggering rejection itself. But to give you an idea, if you are the most serious risk cohort, so your donor is positive, you're negative, you are an older person, you have ATG and delayed graft function, by the end of 10 years, you have very little hope of retaining your graft. So now we talk about evidence-based health policies. What do we want to do? We're building societal groupings using deliberative dialogue strategies to try and engage everybody in the province, including the public, patients, and providers, because we want their input as we go forward. We're beginning to build health economic models of the program in BC that can be used across the country, and these include pre-transplant waiting list times and post-transplant outcomes. We're beginning to think, how will we all share data in the future? The possibility for pooling data between provinces is rapidly shrinking. Provinces don't want to allow data to be pooled. So we're looking at strategies like blockchain, where we can actually set up servers in all of the different hospitals or different universities. We can put our data in there. We can send an algorithm through that will help to analyze the data, then do a meta-analysis of the data. And we're working with Vicky Lemieux at UBC, who is one of the Canadian leaders in this field. So we would set up our server networks here. The data goes out to our UBC blockchain server, then it's shared with other groups. And this is one of our partner hospitals in Germany and the partner hospital in McGill, where we're working on this at the moment. But let me just show you something interesting. We tend to get so focused in transplantation that we tend not to think outside it. But HLA is related to many other things, and one of them is severe adverse drug events. And they often present as severe or devastating and lethal skin diseases. So Stevens-Johnson syndrome and others, picture here of the sort of blistering of epidermal injury that occurs. And if we were able to know in advance the risk gene, the HLA risk gene of the patient, we would avoid providing this, whatever individual anti-epileptic drug, allopurinol or other to them. But of course, conventionally, it takes us 10 days to get out a result with sequencing for HLA. So we're actually beginning to use nanopore sequencing in a new approach, where we receive the blood or the buccal swab from the physician that morning while the patient's still in his office. We sequence it, we send out the results at midday, and we add to that an artificial intelligence assembly of all of the information distilled along with expert opinion to guide the physician to treat. So we're just moving forward on this with another Genome Canada proposal, and we actually believe this will be one of the first in the world. So a very exciting extension, completely unrelated from the field of transplantation. So thank you to all our colleagues, to James, to Karen, everybody who participated, to everybody in Alberta who's been involved in the labs, clinically, and otherwise. Thank you again.
Thank you, Paul, really, for sharing this gigantic wealth of insights and information, right? With being a pathologist, seeing that what the cancer world is doing with this technology to guide therapy, risk stratify patients, I think it's just you have to be applauded that you drove this in the non-cancer world, so to say, around. And as you say, there's many diseases other than transplant, but transplant usually gives us a great learning opportunity. And I'm really impressed how you designed this opportunity to have impact of how we care for patients. So
thank you.
Congratulations. So we have time for a few questions. I'm not sure. I try to monitor this, whether you raise your hand or put it in the chat. So while people get ready for that, one thing we struggle with in antibody-mediated rejection is non-compliance, right? The breakthrough of your immunosuppression and then developing DSA. Did you have a chance to look at that? So you can't predict the risk of non-compliance from your genes. I get this, but is there an increased risk for DSA in non-compliant patients depending on their high resolution match?
So a really great question, Mike. We're actually in the middle of it. So what we have done in preparation is we've gone back through the last 1,500 patients in the cohort. We've asked who develops DSA. And actually, the number is surprisingly small. Of the 1,100 that were fully sequenced and on whom we had samples, only 60 patients developed demonstrable DSA. So 5%, let's say. This is, I mean, it's far, far smaller than we had ever anticipated before. And we're now asking, is that related to the disparity in HLA? I bet it is. I would put $100 on it now, but I can't say with confidence until we know that.
Yeah, it would be. Well, I was at this FDA patient engagement workshop on immunosuppression, right? And often non-compliance is associated with the severe side effects, these very toxic drugs, right? And the amount of immunosuppression we give. And the question was really about, can't we do better and have more tailored drugs? So that was your middle transition of identifying low-risk populations, which we might overtreat. But of course, everybody's afraid when you do not treat them to standard of care that they might develop DSA. And once you have the DSA, you have a problem, right? So I'm just wondering how these data you generate can help us also dealing with compliance challenges, not only whether they are on enough and the right immunosuppression, but on a more compelling protocol, not taking somebody said they took 30,000 pills since they got a kidney transplant, right? So this is all has a personal impact, and maybe these data can help us risk stratify around that as well.
So I think you've described it perfectly, Mike. We have that would be the hypothesis. And now we have the information that will help us to understand, because we have now probably 4 or nearly 5,000 fully sequenced patients and donors we can go back and ask. And then we have the tools for the experimental trials, the short, simple, historically based comparators. So I think we have tools that we never had before, and we can begin to answer these. And the nice thing about it is I think Canada can answer them much more easily than the States, because we can work together easily.
Okay. No, I really hope for that, right? And I agree with you that Canada has this setting to show that it is health economically useful and from a patient perspective, right? In a multi-center setting. I agree. We're positioned for that. So while and another wild question, it might be totally hypothetical, is I'm always wondering the cell-free DNA coming from the graft. What information does that carry, and can you match that to when you have a high resolution type patient to get more specificity of what is going on in the graft, or what is actually truly just I'm always doubting, do we have to do, once you have high resolution type somebody and you know it, do you have to do the standard methods of just saying this is DNA from the donor, this is from the recipient? So is there value in exploring whether cell-free DNA from the graft is a good follow-up tool in this context?
Yeah, I think the answer is yes, but the key question is it predictive? So all of the studies so far are really associative. We see that there is an increase in cell-free DNA in association with rejection, but that's not really helpful, because we would always do a biopsy anyway. What we really want to know is, does it increase over the weeks before rejection that will give us time to intervene before there's injury to the graft? And I think you're probably part of the study that James is just setting up now, which I think is a wonderful study. It's going to be the first one to really look at predictive kinetics of cell-free DNA. And I just don't know whether it will be predictive, or at least will give you enough time to intervene.